Ah. Yeah, I was starting the recorder.
Oh, so it's going in?
Yeah, so that's definitely one aspect. The end game for these machine learning models is to generate systems that sort of clinicians can make decisions on. So you can aggregate all that data and make some decisions that can help improve care for your patients. So today, we're going to talk about machine learning, sort of in the context, sort of define what it is, go through a couple case studies, and just think about where this fits in the continuum of some of the things that you're doing. There are lots of applications. My personal focus is in clinical informatics. So I think about how we take electronic health record data and sometimes combine that with genomic data to predict certain outcomes that are going to happen. And those outcomes can be used to direct our patients' care. So I'm an MD PhD. I'm in the Center for Health Informatics and Bioinformatics. My primary expertise is in machine learning. So both predictive modeling, which is predicting things, and we'll see some examples of some of those things throughout the lecture. Text classification, so taking documents and classifying them into groups, so like spam and not spam, right? So that's sort of a very high-level example of doing text classification. Data mining, so doing social media type of stuff, right? So we build models in Twitter to identify, whether people are using alcohol or whether people are using drugs of abuse. And when you get to that level of scale, there are 500 million tweets per day. You leave the ability to do this manually. You have to have computing systems to support you to make those classifications. And then large medical databases. So there are publicly available databases such as Sparks, which is every inpatient and outpatient admission in New York State. It has, I think, 40 million patients. And so, once you hit a certain scale, you can no longer do this by hand. And you need computing, and you need a computing infrastructure to help you analyze that data. Secondary expertise are building search engines. So I've done that for a couple years and built a system here at NYU to help faculty find each other. So it's called Find a Collaborator, and we can see an example of that if we have some time later. And then natural language processing. So electronic health record data is full of natural language. And so how do we extract relevant data out of that? So I do teach a couple courses, the Intro to Medical Informatics course, the Intro to Medicine for Computer Scientists. So computer scientists typically don't know anything about biology and they don't know anything about medicine, so we provide a course to help them learn about that. And then finally, data analytics in R for physicians. So we teach physicians how to analyze data. So what is machine learning? So let's start with a couple of examples, rather than trying to define some esoteric definition. So these are some of the primary examples. So number one is, if you have an email, is it spam or not spam? That's sort of the prototypical application that first set this entire part of this field off. If you notice, if you use Gmail or Yahoo, you just don't see that much spam anymore, and that's all because of machine learning systems that are built to identify specific words and combinations of words that seem to indicate something as spam. But you may have experienced it as well. There are times that it's not 100% accurate. You have an email that came from someone that you know or someone important, and it slips through the cracks. These systems aren't perfect, but they're better than getting hundreds of emails, of spam, and then having to worry about it. So people are willing to take that trade-off. So we can see here email spam text classification, and we won't really do an example in class today of that. So another example is digit classification. So back in the 1990s. People were wondering, what can we use to classify digits automatically? We can actually do it really well. Some of the machine learning techniques now are really amazing in being able to do this. It's actively used by the U.S. Postal Service. So every day, every time you write a letter, it actually goes through a system that scans the zip codes, and it'll apply these algorithms to it and try to give it, the actual numbers that they belong to. And the ones that it can't figure out actually end up getting triaged to a person who actually looks at them and tries to figure it out. And there's actually a really funny website you can go to where they show all the examples of ones that were not readable, and they are just really, really not readable. So the system's doing a pretty good job. Um, a very simple example that to sort of get a sense of what these things are doing is predicting Titanic survival. So there was a data set published about 30 years, 20 or 30 years ago, where they just captured information about people on the Titanic, and you can see some of the examples there. So there was the passenger class they were in, what their name was, what their sex and age, a number of siblings and spouses, number of parents and children aboard, ticket number, the passenger fare, the cabin, and port embarkation. And they actually had labels for each one. So they knew for each person whether they survived or whether they died. And so the task is to use some sort of machine learning system to basically predict, did this person die or did this person live? And there was a competition to do this, which is in that link below. And people are able to do it pretty well. I think you get like 85% accuracy, I think, with some like even things like logistic regression. So the main components that you see here is that we have features, we have things that describe our data, and then we have a classification task. So in this case, we're trying to classify people as to whether they died or survived. And finally, the more bioinformatics application is sort of molecular signatures. So back in 1999, Golub, who I think is at the Broad Institute now, but this is sort of the seminal paper that sort of kicked off this whole idea of using, machine learning to classify things. So this is a cancer classification. You can see there are two groups, AML and ALL, so acute myelogenous leukemia and acute, can't remember what the other L is, leukemia. And so they had a bunch of genes. Lymphoblastic, yes, leukemia. So what you see here is that each row represents a set of genes, okay? And those are the values that you have on a gene array, for example. And then each column represents the classification of whether it's ALL or AML. And so the challenge was to come up with some system that would do this automatically. So put it in some black box or whatever, logistic regression, whatever you want to do, and see if it can figure out what the right outcome is, whether this person had ALL or AML. And we'll talk about this example a little bit more in the case study, but this just gives you a flavor of, of what the usefulness of this is, in bioinformatics. Yeah.
Kind of along the same lines, is this a similar type of technique that gene sequencing facilities like 23andMe and other large companies using, like based on someone's gene expression profile that comes out of sequencing, they can put in those data and say, oh, you have a X% chance of having this disease.
Yeah, yeah. So I think
another company might have a totally different
right.
algorithm, and then they come up with a different diagnosis based on the same patient sample.
Yeah, yeah. So, I guess for the video, the question is, are technologies like this, in 23andMe, trying to do predictions based on, gene sequences? So I'll give my answer, and then we'll have Stuart give his answer, because I think he probably knows a little bit more than me. I don't think 23andMe is doing anything so sophisticated. I think 23andMe is really focused on sort of single locus types of situations where, okay, we know this gene is connected to Alzheimer's, so this increases your risk by X amount. You could imagine, and we're getting to this. I mean, there is data now where people look at gene sequences and see that, do you have atrial fibrillation or not? And they look and they mine that gene sequence and they come up with SNPs that are related to atrial fibrillation. And then when they go back and look at the wet bench work, they say, oh, look, the top 3 or 4 are actually SNPs that are related to atrial fibrillation. So this is what they do in the health record, right? So they mine the health record for all kinds of phenotypic outcomes, and then they'll go backwards and connect that to the gene sequence. And then they'll do what they call a PheWAS study, so PHEWAS, so a play on GWAS. And they'll actually say, oh, look, for these phenotypes, these are the genes associated with this phenotype. And then either people will go out and verify it on the wet bench, or they already verified it before and had it validated before. Did you have anything to add, Stuart?
I agree with what you said at first, that the single locus predictions of what can be done, and it completely glosses over the combinatorial aspect of it. Yes, more people who have this allele have the disease, but in your personal other factor that's static or blocks it or negates the value of the first one. And there is no algorithm sufficiently complex to integrate all the markers from the genome at this point.
Yeah, and we'll talk about that. There's this idea of the curse of dimensionality, which is once you get into really high dimensions, it becomes really, really hard to do machine learning in really high dimensions. So we'll see an example of that later in the talk. So what is the goal of machine learning? Well, it's really to construct algorithms to learn from data, right? So if you have data, I want to build an algorithm. But what's the purpose of that algorithm? Well, the purpose is to learn from training data so that you can apply it in data that you haven't seen before. So, if I sequence all of you except for Stuart, and I learn something about all your sequences, and I build a model on all your sequences, I want to be able to apply it to Stuart's sequence and be able to, predict an outcome. And I don't include Stuart in that group because I could easily, if I use Stuart to train that algorithm, well, it's pretty useless to just predict on Stuart because I already have data about Stuart. So there's a general framework, in sequencing. The first is you obtain the sequence, and so that can happen through microarrays or whatnot. In a typical machine learning problem, you also have sampling considerations, right? So when I'm trying to build a spam classifier, I think Google probably gets probably, I don't know, 30, 40 million spam, well, new spam messages, probably like in the order of several million per day, right? And so, you can sample that to try to build a classifier. Some of the modern techniques, you can do all of them, actually. But there is a sampling step where, you try to sample in your space to build your model. And you hope that sample is representative, right, of your actual, the actual groups that you're going to be applying this to. Labeling the sequences, so ALL and AML, right? You have the row, you have the information about them, and someone has to go and label it. So a lot of times it happens through the pathology or some sort of gold standard. So someone says, did the biopsy and says, oh yeah, this person has ALL or AML. You have to clean the sequence up, right? Which we get the raw microarray sequences, they can be pretty messy. Maybe some things are not in the range. And so there's a cleaning step, where we actually manually go in and sort of clean up, outliers and this and that. And then that gets it ready for sort of the machine learning algorithms. And when we apply the machine learning algorithms, we do a validation because, we're not really, what we're most concerned about is how well does this model work on things that it has, on sequences it's never seen before. And so we have techniques to internally validate that, right? So I can take the people in this room, and if you had ALL and you had AML, there's an internal way that I can just take the group here and be able to estimate what I think is going to happen when we do it on Stuart or Ross. And then there's an external validation. So one day I'll build the model with all of you, and then we'll go to another class at Sinai and get 20 of their sequences, and then we'll apply our model and see if it works. And then that's the sort of the true external validation to say how well our model actually works.
At what point
Yes.
do you trust your tests of the validation? Like, do you have to have an N of 5 or 30 or 1,000, or is it a proportion of
Yeah.
the...
So, at what point do we, trust our model? And so, a couple answers. Number one is if the signal is really, really strong, right? I'll show you the technique that we use a little bit later. You can have really low sample. I mean, you can have samples in like 50 or something like that, and be able to get really, really good classifier if the signal is really, really strong. Now, if the signal is really, really weak, you may need more sample, right? And this is sort of classical statistical, type of theory, right? In the sense that if you have something, when you go to your statistician, you're calculating sample size, right? You calculate your sample size based on what you think your effect size is. If your effect size is really small, then you need a lot more sample to be able to detect that difference, right? But then if it's really big, you don't need as much sample. So the same rules apply. Typically, I think in the ALL, AML case, they had 71 cases. And so, you could see from our previous picture here that, the signal is actually really strong, right, in ALL. Like, it's pretty obvious on the, or AML, it's pretty obvious on the far right there, that there are genes that are clearly, expressing more. And so in this case, I mean, you probably don't even need 71. You could probably do it with like 10, right? right, and build a pretty valid signal. So it really all depends, and a lot of times we answer this question empirically, where we just run the experiments, see what the performance looks like. If it's not good enough, then we'll do some troubleshooting to see whether more sample is going to matter or not. So the basic framework is we want to take a set of labeled examples, and we want to run a classification algorithm, and then we want to take some to build us a model, and then we want to take some unseen examples, which are shown up there, and actually classify them into the right group, into the right classification. So that's sort of the basic model that we're using. So there are a couple key concepts to get out of doing this machine learning stuff. One of them, this idea of supervised learning. So here's an example of a shoreline. Right? You have a bunch of boats, we have a bunch of houses, and you want to classify the two groups together. And so let's see. Let me ask you. If you were to draw a line that would separate these two groups, where would you draw that line? You got the boats down there and houses up there. Where do you think that line would be? Exactly, right. So in this geometric problem, that's what we're trying to do, is we're trying to come up with sort of what we call a hyperplane, or a line that's going to perfectly separate our data. In this case, we can do that, right? So we have a line on the shore, and it separates it into two groups: the boats and the houses. And the coastline serves as sort of this decision surface, right, to separate them into two groups. But sometimes we get tricked, right? Because sometimes it's not exactly clear, and our line isn't exactly perfect. So sometimes we have boats that are misclassified, or homes that are misclassified as boats, right? So you can see up there, there's some sailboats that are sitting on the shore, right? So those are misclassified if we use this rule. And also we have some houseboats out there too, right? So houseboats are sitting out in the water, and they end up getting misclassified, right? So our lines aren't always perfect, but that's okay. In this case, we want to learn to separate the boats from the houseboats. Making a few mistakes is all right, and we're willing, and we can tell our algorithms to expect a few mistakes so that it draws the lines correctly. So the principle here is, we can use latitude and longitude. Imagine latitude and longitude being two features of your data set, and we can end up drawing a line that can separate the data. In this case, it's the coast. But what we actually want to do is, at some point in the future, we want to have some things that are that we've never seen before. And so if I, have some things that are a certain latitude and longitude, and other things that are certain latitude and longitude, with this line, I can make a prediction about whether it's a boat or a house. And all I have to do is look at the side that it's on. If it's below the line, it's a boat. If it's above the line, it's a house. Yeah, and then we can do the same thing here. So that's what they call supervised learning. As you saw there, the examples are labeled, right? So we have labels for each example. We have the houses, we have the boats. That's what supervised means. It means that you have the labels available, and you give those labels to the machine learning algorithm, and then the machine learning algorithm figures out how to define a surface to separate the two groups. That's in contrast to an idea called unsupervised learning. You may have heard of things like clustering, and clustering is an unsupervised method. So you don't have any idea from the get-go what the classifications are. You're just hoping to put them into groups, right? You let the algorithm just put things into groups. So that's principle number one, is supervised learning. All right, so this part, let me get your attention for just a little bit and really focus for about 5 minutes. There is a very important principle called underfitting and overfitting that we really think about literally all the time in machine learning. The principle is that you can overfit a model if you end up building a model that works good in your original data set, but works really poorly anywhere else. And you can underfit your model if you have a complicated function that describes your data, but your learning algorithm just isn't powerful enough to predict that. So let me do an example. So suppose that we have this set of data. Okay, we have an outcome matrix Y, and we have some predictors X. And so let's say out of this, we actually have some training data. See, the purple lines are little training data. We can build two models. Here's one model. So we can take the training data and we can fit the data exactly, right? So this is a 1, 2, 3, 4, 5, 6. This is a 7th order polynomial, right? So you see it fits the data perfectly, the training data perfectly, right? Another option is to have the straight line, right? So this is just a logistic regression line that we've drawn, we can sort of just draw that in, and then it fits our training data as it fits our training data. So here's my question. I will ask you. So we have two lines there, right? Which one do you think fits the data better? So the circles are going to be the true data, right? And the purple ones are the training data that you give. And these are two solutions to this problem. You have a curvy line and you have a straight line. So which one of those do you think gives you better, which one predicts the circles better? The straight line. Now, why the straight line? Yeah, okay. So it's a straight line. Does anyone have a thought on why the straight line might be better? Yeah. 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 So yeah, definitely. So if the reason we know this is the right answer is that we can do some calculations. We do some maximum likelihood types of estimates when we build this, and we can actually see that this straight line minimizes the error, right, that occurs when you're trying to fit something to this data. So, so what we see here is we see this. This is an example of overfitting, right? So if I take a set of data, I can perfectly model that data with an algorithm of some sort, right? But that's not good enough, right? Because then I end up overfitting data. I create a model that works only for the training data that I have, and once I go out to test it in the test data, it just doesn't work that well anymore. So in this example, you see that this line overfits, but this line is good, right? The straight line is good. So the take-home point here is that I've just told you that if you fit the data perfectly, you build a model that's no good, right? That doesn't generalize well, and that it doesn't work anymore. All right, so let's look at a second example. So here I have another data set. You can see that it looks kind of like a parabola, and I have again the training data in purple. So I'll go over here this time. So tell me. Well, I'll go through this and then you can tell me which is better. So here's one line, right? And it fits the data perfectly, right? Here's another line, just like we did before. It's a green line. It's a straight line through the data. So which one of these is better? The straight line or the sort of curved line? Yeah. Say again? The curved line, right? So, and why is that? It's what? Right, right. So we see here that we actually have an example that is of a set of training data that's actually not represented by a line, right? It's actually represented by a parabola. And you could sort of, put a parabola there if you want to. And so here's an example. So remember, in contrast in scenario 1, I just told you that if you fit the data perfectly, that's a bad model. Well, here I gave you the opposite scenario. If you fit the data perfectly, you actually have a better model than if you just had a line, there before. So the take-home point here is, and this is an example of underfitting, right? So I have a model that looks like a parabola, and I apply an algorithm that the best it can do is make a line. It can't do anything more than make a line. And so I've actually underfit this example because the machine learning algorithm just isn't powerful enough to learn the function that's actually in the data. So then, I end up having this parabola, looking example, and that's what, is actually good data, or that's actually a good machine learning algorithm. So I just showed you an example in scenario 1 where if you fit the data perfectly, it's no good. I showed you an example 2 where if you fit the data perfectly, it is good. And then in scenario 1, I showed you a line that was good, and here I showed you a line that was bad. And so these two interplays between fitting the data perfectly and also fitting as well as, so the interplay between fitting the data perfectly and having a simplistic, having something simple are always counterbalancing each other. And so in machine learning, we're constantly trying to come up with the perfect balance between this overfitting and this underfitting to create a model that's going to generalize outside of the data that we train with. So if our signatures are too complex, this will overfit. And if our models or signatures are too simplistic, it will underfit. Any questions about that? Right. So the next key concept is performance estimation. So earlier I talked about, we can take a data set that we have and internally model what the internal performance of that is going to be on data that's never been seen before. So the way that we do it is something called either holdout validation or end-fold cross-validation. So you can imagine in the holdout validation, if I have 100,000 gene signatures and 20% of them are AML and 20% and 80% are ALL, I can arbitrarily separate into two groups, right? Train on one group and then estimate performance on the test group. And that performance will be what I expect the performance to be if we go to Mount Sinai and do the same experiment. Sometimes I can even get a more exact estimate by doing something called end-fold cross-validation. And you can see that in the bottom example. So I have a set of data, and I divide it up into, in this case, 6 different folds, right? 6 groups, where each of the test groups, if you sum them all together, would be the full data set. Okay? And you can see that the test group is different in each one. And so what I would do is I would go to the first group, I would train a model on the first, and then apply a value to get a test result, generate a result for that one. Then I would go to the second one, build my model on the train set, test that model, get a result 2. And one simple thing I could do is just do all 6 runs, average the performance on all 6 of them, and then that's my generalization performance. That's the performance I would expect to get if I went someplace else and it had exactly the same, example, and it was for exactly the same problem. That's the performance I should get if I go, to some other university or some other set of patients to do this. Yeah.
So since you have so many different training sets,
Uh-huh.
what do you consider
Yeah. So once you've estimated performance, right? So we do this just to estimate performance. Once you finish, you need to build a model, a final model, what we call a final model, that can be applied, to Mount Sinai that will use all the data, right? So there's another version of this called nested cross-fold validation, nested end-fold cross-validation, which I won't get into here. But what it allows us to do is to optimize the underfitting and overfitting of the algorithm, to find that optimal overfit and underfit parameter, and then build one model out of all the data. So there are techniques certainly to do that. But usually, we can only evaluate performance with the data that we have. Usually the external test case is really expensive. You got to get all these people, to agree to help you and this and that. So, we use this technique to be able to get a general sense of, okay, well, if I actually tried to do this at some other place, how well is it going to work? So there are other versions of this that you may have heard before. One of them is called leave one out cross-validation. So in the example before, we left a group out. Well, there's another example where you can leave one out. So, I could take all of you, leave Stewart out, build a model, apply it to Stewart, get a result. Then I could do everyone except Ross, build a model, test it on Ross, get a result. And do that over and over again for all of you. So that's what, like 20 times? And then average the result of those 20 times. That would be my estimate of performance outside of the data set. Now, the advantage to doing that is I get to use all the data, right? So I get to use all 20 of you before I do it on that one person. Whereas if I divide the data into 5 folds, right, I'm actually only training on 16 people and then 4 of them are part of the test set. So by doing leave one out cross-validation and samples when I have really low sample size, I can maximize the amount of data I'm using to train. There's another example called leave pair out cross-validation, which is leaving pairs of people out. There's the bootstrap with replacement. So there I just randomly select, 15 of you with replacement, build a model, and apply that. Unfortunately, the estimated results of that are not stable if you try to do it that way, but I just put it up there and take part of it. And then the single holdout, which is the example that we saw before. So any questions about leave one out cross-validation or how we estimate performance? Okay. So one of the most popular algorithms, machine learning algorithms out there is something called the support vector machine. And it's really based on the example that we gave earlier of looking at the latitude and longitude, looking at the homes and the boats, and it's a geometric procedure that tries to find an optimal splitting of the data. And so we'll look, and we're just going to go, and look at this one example. There's actually, many. There's, I don't know, I wouldn't say hundreds, but there's lots of different machine learning algorithms out there that all have their different strengths and weaknesses. This one is especially nice because of a couple reasons. Number one, it works really well in very large feature sets. So if you have 50,000 features, but you only have like 100 patients, this is your best bet of getting a model that's actually going to work. If you try to run logistic regression as classic logistic regression on it, it wouldn't work. The numeric, trying to inverse the matrix for the logistic regression to find the right answer is really computationally extremely expensive, and usually you run out of space and it doesn't work anymore. You can learn both simple and complex models. So earlier we saw an example where you had a straight line, we had that straight line, we had the parabola. Well, this thing can actually learn a parabola if your data looks like a parabola. So that's pretty cool because now you're no longer, limited by the linearity of the data and what it may look like. And finally, it uses some certain principles, which I won't go into here, to avoid overfitting. And it works really well empirically. So suppose we have this example. I have gene X and gene Y, have some circles, and have some stars, right? So there's gene X and gene Y, and I really want to try to find, the perfect line, to separate this. We do have. So here, I'm going to take this off for a sec and go to the board so that people who are listening don't get the benefit of this. So let's suppose that we have. So let me ask you, if I were to separate this data, where would what line, what could I draw to separate this data? If I were just doing lines. Yeah, so in the middle. So where in the middle? Or do you want to come up and just draw a line? Come up and draw a line. Okay, so that's one line. So absolutely. So now we can, so this line is perfectly valid. It separates the data just as well. So does that line, right? Would anyone argue with me that that line is just as good as this other one? And then I can keep on doing this, right? I can even get really close like that. So the thing is that there's actually one line that's better than every other line. And you can sort of see, if I were to compare this line that I drew right here, this line right here, you might have an intuitive notion that this line is actually worse than this line, right? Because you can imagine, well, what if there's an example that kind of pops over in the same space over here, right? And now, the line isn't any good anymore. So, nicely enough, in 2 dimensions, it's like, okay, whatever, it seems like kind of a sort of problem. But sometimes when you hit, 50,000 features, right? You have 50,000 dimensions on this sucker, right? You need some theory to drive to tell you, okay, what's the most optimal line? What's the most optimal plane that will separate these 2 groups? And luckily, we have some guys who are extremely smart that aren't me, and they've determined how to answer that question. And that's what you can see here in this picture. So this is called the maximal margin gap. And so you can see there's things called support vectors. Those are in yellow right there, the star and the circle. And that line that's represented in the middle, that maximizes this gap, you see that gap up there, that's actually the best line that you can possibly make for this problem, given the data that you have. And so the goal of the support vector machine is to actually find this line and to maximize that gap. So the cool thing about SVMs as well is that it can learn some crazy stuff, right? It can learn even things that are not linearly separable. So imagine I have a data set like on the left, right? I have X and Y, and I have you know, basically all my squares are right in the middle of all the circles, right? So there's really not a function, you know, that could, not a linear function on one plane that can actually separate that. Well, I can do something really cool. It's called the kernel tree, where I can actually project, and you see there on the right, I've actually projected the data into a third dimension, right? And so now I've actually lifted all the squares up. I've actually pushed all the circles down, and now I can create a separating hyperplane, you know, between those two groups. And the really cool thing too, and I'm going back to the board, is I can actually project it backwards. So after I get it to this state, so once I go back into, you know, so once I've created this realm here, the cool thing is that once I do it in this kernel, what we call kernel space, I actually go back into the 2D space, and I actually have a line, a circle, actually that's created, and this will perfectly, you know, this perfectly separate the data. So this is another reason why SVMs are really powerful, is because it gives you the capabilities to learn a function. It gives, it can learn any arbitrary function if you give it the right parameters. And a lot of times with our data, we don't know what the right function is. We don't know if it's linear. We don't know if it's exponential. We don't know if it's a parabola. We don't know what it looks like. So we want to preferentially use algorithms that can learn any possible function. And this is an example of one. All right, any questions about the SVM? All right, so the next concept is the curse of dimensionality. And this is one that, you know, as WebBench and Bioinformatics people, this is one that, you know, it should be always in the back of your mind. So what is the curse of dimensionality? So, you know, let's look at this example. So you see this example before. You know, we start with a single feature, right? So we just have a single feature x1. And we have three classifications. We have circles, we have squares, and we have triangles. We see that you know, we can't actually separate perfectly those groups, right? I mean, we could separate these circles, right, because they're kind of off on their own. But we have a problem because we can't actually separate the squares, you know, from the triangles. A couple things to notice is, number one, the density, right? So you see there's three examples in every block, in every bin, right? There's 3 bins there, there's 3 examples in every bin. So you say smartly to me, Yin, you know, I think we could solve this problem by just add another variable, right? So instead of x1, we're going to have x1 and x2. And so let's do that. So now I see I have x1 and x2, right? And so see what happened to the problem here. So in 2-dimensional space, you know, so I've sort of increased the size of the matrix that I live in. Now, recall in the last example, I had a density of 3, right? It had 3 per bin. Well, if I want to maintain 3 per bin, I have to actually sample 27 points now, right? So I've just had an exponential increase in the number of samples that I need to have to be able to model, to be able to maintain the density of what I had before. Similarly, the other thing I could do is I could just maintain the number of examples I have. And what you can see there on the right-hand picture is that everything's really spreads out, right? So notice in this example how close everything is together, right? So the circles, the squares, and the triangles are really close together. Well, if I maintain density, I get a really dense space. I have to, you know, triple the sample to be able to have the same sample size. Or I get the instance where everything gets really spread out. So you can see that once we go to 2 dimensions. If we go to 3 dimensions, the problem gets even worse. So here you can see 3 dimensions, right? Well, if I were to maintain the density of 3 per box, I have to do 3 to the third to the third. And that's how many examples that I would need to fill in 3 in every box. So I think that's 27, so 27 times 3, so 81 points. That 81 examples just to go to 3 dimensions. Or I could keep it really sparse. I could stay with my 9. And again, you see here how everything's starting to spread out, right? As I go into more and more dimensions, things start to really spread out and get more and more what we call sparse. This problem gets... Yeah.
So you're going to increase dimensions. A number to place each of these dots in each of the dimensions. Where are these numbers coming from? These measurements that you're making?
Yeah, so in this case, you can imagine being 3 genes, right? So,
you're just adding genes.
yeah, so I'm just adding additional genes.
Cool, no problem.
Perfect. So, so you can imagine about 50,000 of these, right? Like, the points are just basically in this space all on their own, somewhere, and they're not even close together. So things that were close together in 2D space, right here where the circles are close together, once I get to sort of, infinite space, they're actually arbitrarily really far apart, even though they seem close in one-dimensional space. And if I do this for, like, hundreds of thousands of features, the mathematicians tell me that all the values eventually end up on basically the rind of an orange. So everything is arbitrarily far away from each other, and all your data points are far away from each other. So here we'll see some of the solutions, to this problem, right? Because if we didn't have a solution to this problem, machine learning wouldn't work at all. Because once it got to higher spaces, it wouldn't be able to function. So how do we deal with this? Well, a couple of things. Number one is we can incorporate prior knowledge, right? So you have an expert that comes by and says, gene X, Y, and Z, those are the important ones, included in your model. And so once you do that, you've gone from 50,000 down to 3. And actually the Golub people, that's what they did too. They had a huge gene array. They selected, I think, 50 of them in their first experiments, and they tried to build their models on the 50. We won't talk about two, but you could also reduce the dimensionality, right? So rather than having, you can have automated techniques that will take the 50,000 and reduce it down to like 100. And that's another way of dealing, with the curse dimensionality. So the takeaway home, the point is though, is that what it does is what it says is that for a given sample size, if you have a given set of sample, there's a maximum number of features above which the performance of classifier will degrade. So if you have a set of points, if you have a set of examples, you have a set of patients, there's only so many features that you can consider before your model is just not going to do that well. There's only so many genes you can consider before your AML and ALL just aren't going to classify that well. And this is really, really serious issue, right? We have gene expression arrays that have 10 to 50,000, features. We have exon arrays and tau microarrays and SNP arrays have greater than 500,000. We have proteomic data sets have 10 to 30,000. Next generation sequences have greater than 100 million. So we really have to, this curse dimensionality is very real. It's something that we have to worry about and think about. Especially in small samples, in your lab, you may work with samples where you have, 20 patients, 50 patients, and you have 50,000 features. This is, a real issue in that you can easily overfit your data. You can easily build models that work great on your data but don't work anywhere else. And especially, there's classical methods that won't work at all. So classical regression won't work. There's some other methods that give bad results. The analysis can be very slow. And it tends to overfit, because arbitrarily by chance you're going to find a correlation, even though there's not a direct connection to that. So one of the examples I like to give about this high dimension and high dimensionality and having an outcome of interest is, are there any football fans in here? Does anybody follow football? Football. We know football, right? So there's the AFC and the NFC, right? They play the Super Bowl at the end of the year. And the AFC and NFC are 2 divisions, right? So all the teams are split into divisions of, I think, 16 teams each. Well, if you look at the data and you look at the people that, if you look at who won the presidential election in the last like 50 years, and you look at whether Republican or Democrat won, you can actually look at the AFC champion, who won the AFC championship, and that's almost a perfect predictor. It's like 85 or 90% accurate of who won the presidential election based on who won the Super Bowl. So that's just a very simple example of when you consider all things.
Wait, explain that. Like,
Yeah.
the AFC, like which team?
Yeah.
Would indicate to are there like Democrat teams and Republican?
No.
teams, or-
It's not connected at all.
But there's more.
It's just if you
teams
look.
than there are presidents.
Right. No. So if you look at the presidential elections, right, and there's a certain outcome for each election, right, and you just go look for any correlation at all, anywhere in the world, of something happening where the Republicans won, this year, and the Democrats won that year, you will find something where that matches.
So it's not actually.
No, it's not.
Okay.
Right. And that's the whole idea behind this high dimensionality, right? If you have 20 patients, you have 50,000 features, by chance, you're going to likely find something that seems relevant, even though it's not. And that's where all the statistics that you guys are learning come in, where you think about p-values, right? And you don't want things to happen by chance because you want to control for that type of, just superfluous by chance outcome. So we will finish the rest of the lecture looking a little bit more closely at Gallup with some of the principles that you've learned. And then we'll talk a little bit about molecular signatures. So for some of you that are working in the wet lab, the end game for some of these, for getting to understand some of the pathways and all that is, what clinical usefulness are we going to get out of learning some of the things that we're learning? So in this case study, it's exactly what we had before. We saw those ALL and AML cases. So we want to classify the values of a gene microarray according to leukemia type, AML or ALL. The task has 72 samples, 47 are ALL, 25 are AML, and 5,327 genes. So the first thing you want to do is label them, which we've done. Someone did a biopsy, they looked at the result and said what it was. We want to encode the microarray, right? So we just get a bunch of values, right, for the intensity of each of the spots in the array. So the first thing we're going to do is within each training fold, right? So we're thinking about performance estimation, right? We have that train and then test. Within each train, we want to normalize the values of each column between 0 and 1. So for all genes, all the values, we're going to normalize it between 0 and 1. And that's to help the machine learning algorithm sort of not have to deal with really outside of the scale values. Notice that we don't normalize across the entire data set first. We just normalize within the train set only, right, and then test it. And this whole principle goes back to, you don't want to mix in your test data with your train data, right? You want to keep your train data separate from your test data. Because the moment that you use any part of the test data to do any sort of performance estimation, you've essentially overfit, and you're creating a model that won't work well in real life. So we have the support vector machine that we're going to use here. We can, draw the good line with the nice gap, as I mentioned. These couple examples maximize the gap between the two groups. We can also build a model with something called k-nearest neighbors. So this is a little example of another machine learning algorithm. And all we do is we go on this 2D space. If I have this unknown record, which is represented by the circle, and I just look at the things that are closest to it, and that's the label I give it. So in this case, that circle would be a plus, okay? Because I just look at the things closest to it. If you've been paying really close attention, you'll realize that this doesn't work once you get into too high dimensions, right? Because that curse dimensionality thing that I mentioned. So once the dimensions get really high, everything's arbitrarily far away and everything's arbitrarily close. This actually doesn't work anymore in really high dimensions. But in low dimensions, this is another method that we can use. We can just look for what's close to it. Yeah.
So by nearest neighbor, you mean like that box would be a microarray, and then
So
some of the points.
yeah, so the X and Y would be genes. So you could it could be exactly geometric, like in the previous example, right? So you can have that X-axis, which is the value of gene 1, the Y-axis, which is the value of gene 2, and then you have where the plot is, right? So all the pluses are ALL, all the minuses are AML, and then you have a new example where you have the expression level for gene 1 and gene 2. You just plop it down in there, and you look at the ones that are closest. So in this case, K is 3 because we're looking at three that are closest, and then you just give it that example. You can just give it that value. Another machine learning model that's inspired supposedly by how our brains work, but it's not exactly the case. It's something called a neural network. Right? So you have inputs here on the far left. You have a bunch of hidden nodes that are in the middle, and then it produces an output. Right? So for your neuroscience folks, right, this should be the same lines of like axons and dendrons, and like you have inputs coming in, and it releases acetylcholine, I think, and it binds on the other side, and things, and that's what this represents. So there's inputs. The inputs would be the values of the genes, for example. So if I have 3 genes here, I have the values of each one, and then I have the output. So if I have these 3 genes, I want the top output to be AML and the bottom of the output to be ALL. And what the neural network is tasked to do is to find the weights in the middle nodes, in those hidden layers, such that the output is going to be the right answer, such that the output is going to be ALL or AML based on the inputs that are coming in. So these are called neural networks. So this is another machine learning example, or another machine learning model. To do this, we're going to use n-fold cross-validation, like we talked about earlier, and that's how we're going to estimate performance. And these are the performances that we get if we run this experiment. So there is a baseline performance. Right? The baseline performance is based on the proportion of AML to ALL. So 47 to 25. So the simplest model I could possibly build is just to say everything is ALL. Right? So then I get 25 wrong. And that's sort of the baseline. I can't, if I do worse than that, then that machine learning model is terrible and I should just get rid of it immediately. So that's the baseline, it's 65%. It's the proportion of correct classifications. And that's the metric that we're using. So 65%. And so what I can see here is that if I take the support vector machine, I take k-nearest neighbors, I take the neural network, and I build these machine learning models, I can get different performances. So I get 91.7% with the support vector machine, 87.9% with the k-nearest neighbors, and 84.7% with the neural network. So that's an example of using some machine learning technology, some different models on a classic cancer classification task. The actual use case for this would be, patient comes in with leukemia, and that's a little bit contrived because right now the pathology is pretty good. I think the pathological differences between ALL and AML are quite distinct. Is that right? Yeah. So they're quite distinct, so they don't really need to do gene sequencing to actually put them into 2 classes. But at the time, this was, just awesome work because no one even knew if you could just take the genes and figure out, which type of cancer they had. There are lots of uses of machine learning in all parts of bioinformatics. I don't have time to go through all of them, but happens in evolution, text mining, proteomics, genomics, systems biology, microarray, and then, of course, the catch-all other applications. So you can sort of explore and look a little bit more closely at this, when you're at home. But there are lots and lots of uses, and if you Google for machine learning in bioinformatics, or you go on PubMed and do machine learning bioinformatics, you're going to see a host of, all kinds of different problems that people are trying to solve using these techniques and using the basic framework that we talked about. Yeah.
Do you consider building a hidden model a machine learning method?
Yeah, I'd consider it a machine learning method. Sure. I mean, because you're predicting sequences. You're trying to predict the sequence, and it's a prediction task. It's using computational methods to do that prediction. There's a training set. Right? It's training. There's a test that you're applying it to. So, really the algorithm, it's It's less so about the algorithm and more so about the task that's being solved. So the task is you have a training set, the task is you have a test set, the task is you have some mapping of that training to the test, and that's all the algorithm does. So to finish, I just want to talk about molecular signatures. And I think it's important to just have these in the back of your head, because one day when you go see your clinician or you come down with something worse, these are the things that are going to be part of your treatment package. So what is a molecular signature? Well, it's a computational or mathematical model that links high-dimensional molecular information to phenotype or other response of interest. So, make it a little more concrete. So we have a patient with lung cancer. Okay? We want to determine if it's primary or if it's going to be a primary or if it's a metastatic. It's going to be metastatic. So what we can do is we can take a gene expression profile and we can create this molecular signature. It's another way of saying a model, right? We can create this molecular signature to determine whether it's a primary lung cancer or whether it's metastatic. There are several uses. We can model disease phenotype. So for a cancer, we can say exactly what it's going to do, what the diagnosis is, what the prognosis is, what the personalized treatment is. And then we get some ancillary benefits too. So we get some biomarkers for diagnosis. So once we run this, we can find 5 or 6 genes that seem really important. We can go after those in terms of studying them or understanding them more. And then we can discover structures and pathways and mechanisms, right? So people use machine learning for exploratory methods. So they're not exactly sure which genes are associated in AML and AOL, so they'll run the study, they'll find the genes that are most important or most predictive, and then someone goes out and tries to put that in the pathway, right? They try to figure out, okay, what pathway of the disease is this gene associated with? There are quite a few molecular signatures available for patient care. These are some of the different companies that are doing stuff. Agendia, Clarient, LabCorp, Genomic Health, University of Genomics. So let's talk a little bit more specifically about specific molecular signatures available. So here's one for prostate cancer. And what you can see here is that they're all at different points in time of the patient care. So you can see in the top row, you have screening questions, right? And here they want to determine, let's see, what are they determine? So whether you have the disease or not, right? So you see 3 examples there. So after you get the biopsy, then they want to say, how aggressive is your tumor going to be, right? So then they have these 3 signatures that you can just get a sequence and it'll determine how aggressive your treatment's going to be. If you get a negative biopsy, but your PSA levels still stay high, do you need to do another biopsy? Okay. And so there's these tests that you can run that will tell you what you need to do. And finally, after surgery, do you need to do additional treatment if the pathology shows risk after surgery? So you take the prostate out, you check the prostate. The pathologist says, this is pretty aggressive. Do you need to do anything more? And so there are people selling these things, to be able to predict whether you should do something. MammaPrint is another example. It's developed by Agendia, and this is to stratify women with breast cancer that hasn't spread into low risk and high risk categories for occurrence of the disease. It was validated in 1,000 patients. It was so far performed in 10,000 patients. This is like 2 or 3 years ago. The test is about $3,000, and the FDA has cleared it for marketing. And in 2007, this was sort of one of the inventions of the year. So you can imagine how powerful that is, right? If you have a breast cancer to come in, you get it taken out, but then someone could actually sequence you. And I can actually run this molecular signature and say, you know what, you're still in a high risk category. Maybe we should do something. Maybe we should do some. Well, they do radiation anyways, but maybe there's additional things that we need to do. Oncotype DX is another breast cancer assay. It's a 21 gene signature to predict whether a woman with localized ER-positive breast cancer is at risk of relapse. Validated in thousands of patients, 200,000 tests a couple years ago, so probably close to $1 million now. The price of the test is $4,175. It's not FDA approved, but insurances will pay for it. Yeah. So all of these are validated one way or another. There are papers associated with each technology that gives you a sense of what the false positive rate is, what the true positive rate is. And finally, to get FDA approval, those guys really care about, that you're selling something real and that you're selling something that has certain performance metrics. So to actually, once you see that something is FDA approved, I mean, you can sort of, not you can rest easy and be like, oh, it works, but it works up to some standard that they've come up with. Also, none of these tests is going to be ever, there's no test that's ever really 100% accurate, right? So another way to think of these tests is the same thing like, a flu swab, right? You go in, someone swabs your mouth or whatever, and you do, they put it in a little vial, and they do the test. It's exactly the same thing. It's just a test where someone says that there's a certain sensitivity, right? There's a certain true positive rate, there's a certain false positive rate, and the result you get back, it could say that you're negative, but you may be a false negative, right? And so you actually have the flu, even though it says you don't. And so we're always constantly fighting this battle in clinical care, where we run a test, we get the test back, and then we interpret that test, but then we look at the clinical situation, we say, well, the patient still isn't doing well, but the test says it's negative, right? How can that be? And so then, we'll go back, and either do additional testing or just do treatment based on our clinical suspicion. And so we always take the totality of all the information we have to make decisions. And these are especially nice because they help us to make decisions that, before this are just stalled. You're just sort of like, oh yeah, I see a lot of patients like this that, seem to have this outcome. So it gives us a little bit more concreteness to our decision making. So, is it worth it, right? Is it economically valid to do these molecular signatures? And the answer is a resounding yes. So, in the ER positive, case for recurrence, if you ran this thing, if you use the model, you reduce your overall costs by $155,000. And you increase your quality adjusted survival by 16 years by using these tests. And additionally, if you adjuvant chemotherapy with the Oncotype DX assay, you would save approximately $1,930 per woman tested, given a reduction, a 34% reduction in chemotherapy use. So these things are the real deal, people really care about detecting cancer, knowing what it's going to do earlier. And insurance companies are just chomping at the bit to pay for these things because they know farther down the road, they're not going to have to pay for something else. And then here's a couple more. Oncotype DX colon cancer assay, multi-gene signature to predict risk of recurrence in patients with stage 2 colon cancer. $3,280. It's not FDA approved, but insurance will pay for it. Prostate cancer assay, $3,820. Again, not FDA approved, but most insurances will pay for it. Most importantly, Medicare will pay for it, which is what people really care about. And then if we just look at Oncotype DX, this is a little bit old, but in the third quarter of 2010 and 2011, they had an increase of about 18%. And I think recently it's still increasing. And so, there's definitely a demand and definitely a market, for these things. So, as you're doing your research, and thinking about the pathways, and thinking about the things that you're trying to discover, at the end of the day, doing these things, we want to learn more about the biology so that, we can have a clinical effect, somewhere down the road. So this is one of the examples of technologies that exist that where people are using these things to make decisions. So in conclusion, we looked at some machine learning examples. We went over some key concepts. So we talked a little bit about supervised learning, learned what that was, overfitting and underfitting, right? So you can fit the data perfectly, but you have to balance that with not fitting the data perfectly. We looked at an example of one machine learning algorithm, the support vector machine. We talked briefly about k-nearest neighbors as well as neural networks. We talked about cross-validation and how we can use that to estimate performance with the data set that we have. And finally, the curse dimensionality. Right? So when we have one dimension, it's not too bad. 2 dimensions, everything gets sparser. 3 dimensions, everything gets more sparse, and it gets worse from there. And then, having to deal with that with the data sets that we have. And then we look at cancer classification and molecular signatures for case studies. that's all I have. I clearly have time left over. Are there any questions? I see that I've explained myself perfectly, and I try to do this every time. Because if there are no questions, I give myself a pat on the back. Yeah, so if you ever encounter this stuff and want to learn more, there's actually amazing resources online for machine learning. There's tons of free courses that you can take actually about machine learning. So if you never feel confined by actually having to take a course, in an actual real course environment. And because it's a computer science topic, there's literally like an infinite number of web pages and blogs and courses and stuff online all about this stuff. Yeah, so thank you very much for your time. Thanks for listening. All right. So